Assalamu alaikum. So today we are going to discuss the metabolism of carbohydrate. So in metabolism of carbohydrate, we will discuss glycolysis first, then link reaction. After that, T.C.A cycle. Then the product of all these three reactions is used up in the oxidative phosphorylation. We're gonna discuss them step by step, one by one. So we say the ultimate source of energy is light. So during the photosynthesis, light energy is stored in the form of potential chemical energy in carbohydrate, glucose, in carbohydrates, especially in the form of glucose, lipids, and protein molecule. We say these three major organic compounds, carbohydrate, lipids, and protein, they contain the potential chemical energy. So this potential chemical energy is supposed to be released from these compounds and used up in a different activities inside the body, like active transport, cell division, protein synthesis, where the energy is required. But this energy cannot be used up directly, so it's supposed to be converted into ATP. So energy is converted into ATP, then it can be used up for the muscle contraction, cell division, active transport, and various energy requirement processes inside the cells. So we say, first of all, we start with the glycolysis. Glucose molecule contain the potential chemical energy. That potential chemical energy is released. So we say in the release in the three major steps of glycolysis. First step is known as phosphorylation of glucose. Second step is known as splitting of hexose into one bisphosphate molecule. And third step is oxidation of triose phosphate. So these are the three steps which are used which are used to break down the glucose molecule and ultimately into the pyruvate molecule. We would discuss them one by one. So we start with the first of all. Now we say these are the three major steps which are used in glycolysis. First step is known as phosphorylation of glucose. During the phosphorylation of glucose is attached with two phosphate molecules. So glucose molecule is converted to the hexose 1,6 bisphosphate. So first of all, we should know the glucose molecule enter into the cell. So this one is a glucose molecule. It will enter into the cell. Inside the cytoplasm of the cell, glycolysis would take place. So for the entry of the glucose into the cell, a special transport protein is required. These are known as glucose transport protein. We also use the word GLUT for this. Through the glucose transport protein, glucose would enter into the cytoplasm of the cell. Inside the cytoplasm, glycolysis would take place. So these three are the major steps of glycolysis. So once the phosphorylation of glucose, after the phosphorylation of glucose molecule become polar, it cannot come out of the cell. So it stays inside the cell. So it is used up in the process of glycolysis. We start with the process of glycolysis. During the first step of the glycolysis is phosphorylation of glucose molecule. In the first step of phosphorylation of glucose molecule, glucose attached with one phosphate to give us glucose 6-phosphate. Glucose 6-phosphate is further isomerized to convert into fructose 6-phosphate. So we say this one is attachment of one more phosphate will take place. So will give us fructose 1,6 bisphosphate. So first of all, phosphorylation of glucose will take place. As a result of this phosphorylation of glucose attaches with two ATP molecules. So right over here, instead of gaining the energy, instead of making the energy, so two ATP molecule would be used up to give us hexose 1,6 bisphosphate. Hexose 1,6 bisphosphate is a 6-carbon sugar. It is attached with two phosphate molecule. Right? So right over here, glucose used up two ATP molecule. So give us hexose 1,6 bisphosphate, means 6 carbons attached with two phosphate. One is attached at carbon number 1, other one is attached to carbon number 6. So we say first step is phosphorylation of glucose, and the next step would be splitting of hexose 1,6 bisphosphate. In the first step, hexose 1,6 bisphosphate was formed. Now this hexose 1,6 bisphosphate would split into the two molecule of triose phosphate. How many molecule? Two molecule of triose phosphate would form. Now these two molecule of triose phosphate, known as glycolyl 3-phosphate, other one is known as dihydroxyacetone phosphate. So these glycolyl 3-phosphate and dihydroxyacetone phosphate, they are interconvertible. Like say one can be converted to another one, but they use it for the use in the next step, glycolyl 3-phosphate will be used up. So we say first step was the phosphorylation of glucose. And the second step is, like say, as a result of phosphorylation, hexose 1,6-bisphosphate is formed. Now this hexose 1,6-bisphosphate would split into the two molecule of triose phosphate. Triose phosphate means right over here, each carbon compound was, like say, each compound was containing 6 carbon. So right over here, it contains, each molecule is contained three carbon atom and one phosphate. So we use the word triose phosphate. Right now, this triose phosphate is formed. So there, like say, in the third step of glycolysis, there will be oxidation of triose phosphate, mean oxidation of glycerol di 3 phosphate. So during the oxidation of glycerol di 3 phosphate, so coenzyme NAD is required, dehydrogenase enzyme is required. So oxidation of triose phosphate, it would give us, it was during the oxidation of triose phosphate, so it would give us two pyruvate molecule, or let's say, as well as it would give us two ATP molecule and two NADH molecule. So we gonna give, like say, study them one by one. So right over we can say, during the oxidation of triose phosphate, as a result of oxidation of triose phosphate, so will give us one, like say, we will give us, like during oxidation will give us one NADH, like say two NADH are formed because this reaction would take place twice, and like say during the each reaction two ATPs are formed, so ultimately will give us, like say, two ATPs are formed because there will be phosphorylation of two ADP during the each reaction. So we say, now we're going to link them one by one. So first of all, so these were the steps of glycolysis. If we consider the stages of glycolysis, during the stages of glycolysis, the first stage is phosphorylation of glucose, which would take place in the first step. So if you have to, like say, if you can have two scenarios in the examination as well. You can have the question stages of glycolysis, or you can have write down the steps of glycolysis. When the examiner say write down the step of glycolysis, you are supposed to give us these three steps. First of all is oxidation. First of all is phosphorylation. Second one is splitting of hexose 1,6-bisphosphate, and third step would be oxidation of triose phosphate. But if you have the question, write down the stages of glycolysis. During the stages of glycolysis, you are supposed to write down these 6 stages one by one. So first stage is, so these stages and steps, they are interlinked with each other. We can say steps are explained in the stages of glycolysis. We say phosphorylation of glucose would take place. So right over here, during the phosphorylation of glucose, one ATP would be used up right over here. Right, so glucose would convert into glucose 6-phosphate. After the conversion of glucose 6-phosphate right over here, now this glucose 6-phosphate, after the conversion of phosphate to the glucose, so this glucose molecule become activated, it can be used up in the glycolysis. So right over here, glucose 6-phosphate would convert into fructose 6-phosphate. So glucose and fructose, they are isomers of each other. So the next step would be isomerization of glucose 6-phosphate to fructose 6-phosphate. Now we say this fructose 6-phosphate would be further phosphorylated by using one more ATP. So we say fructose 6-phosphate would convert into fructose 1,6-bisphosphate. Fructose is a 6-carbon compound. So one, like 6-carbon compound, one phosphate is attached at carbon number 1, another one is attached at carbon number 6. So we use the word fructose 1,6-bisphosphate. So far we used up 2 ATP instead of making the 2 ATP. Now the next step would be cleavage of fructose 1,6-bisphosphate. This fructose 1,6-bisphosphate is a 6-carbon compound. It would, like say, it is broken down into two molecules. One of them is known as glycerol di 3 phosphate and dihydroxyacetone phosphate. So these are known as trioses. So we say during trioses, the third step of within glycolysis, during triose, let's say these trioses, they are oxidized. Oxidation of trioses will take place. So during the oxidation, glycerol di 3 phosphate is converted into 1,3-bisphosphoglycerate. So right over here, during the conversion of glycerol di 3 phosphate to 1,3-bisphosphoglycerate, one inorganic phosphate will attach with one inorganic phosphate will attach with glycerol di 3 phosphate. So right over here, ATP is not used. So this phosphate is known as inorganic phosphate, which is already present inside the cytoplasm. It attaches, so will give us 1,3-bisphosphoglycerate. Right. So this one is known as PI, inorganic phosphate, which is already present inside the cytoplasm. So it attaches with glycerol-3 phosphate to give us 1,3-bis phosphoglycerate, mean this triose would consist of 3 phosphate molecules. So right over here, when the one with phosphate, so same time, one phosphate is attached, so same time two hydrogen molecules are removed, so will give us NADH2. So we, right over here in the step 5, 1 and 2, let's say one molecule of NADH2 is produced. So and then as well as 1,3-bis phosphoglycerate is produced. So right over here, 3 carbon compounds attached with 2 phosphate. So one of the phosphate is removed. So this removal of phosphate, so the phosphate is which was removed, which is going to be removed from this, like say glycerol, this phosphoglycerate is taken up by the ATP. Now we can say we have right over here, we can say in the step 5, we are having the compound glycerol 3 phosphate. Which compound? Glycerol 3 phosphate. This glycerol 3 phosphate dehydrogenase and coenzyme right over here. So they would convert. So we can say stages of glycolysis comprise of these 6 steps. So we're gonna discuss them, these 6 stages of glycolysis one by one. So first stage is phosphorylation of glucose. So 1 ATP is used up over here during phosphorylation of glucose. So this glucose is converted to glucose 6 phosphate. After the conversion of glucose to glucose 6 phosphate, it is activated now. So we can say now it can be used up in the further process of glycolysis. The next step would be isomerization of glucose 6 phosphate to fructose 1 to fructose 6 phosphate. Mean glucose and fructose are isomers of each other. Definition of isomer is so they have the same molecular formula but different structure formula. The next step would be phosphorylation of fructose 1 fructose 6 phosphate. So as a result of phosphorylation of fructose 6 phosphate, one more phosphate is attached with the fructose to give us fructose 1,6-bis phosphate. So right over here, 2 ATP are used up. So one ATP was used in the beginning of phosphorylation of glucose molecule. The second ATP would be used up for the phosphorylation of fructose 6 phosphate to give us fructose 1,6-bis phosphate. Now this fructose 1,6-bis phosphate is 6 carbon compound. It would split up into the three molecule of trioses. So fructose, cleavage of fructose 1,6-bis phosphate would take place. So as a result of this, glycerol 3 phosphate is produced. Another molecule is known as dehydroxyacetone phosphate. So now these two glycerol 3 phosphate, dihydroxyacetone phosphate, they are interconvertible. They are interconvertible molecule. So now over here in the stage 5, glycerol 3 phosphate would convert into 1,3-bis phosphoglycerate. So glycerol 3 phosphate is a is a 3 carbon compound. So it is again converted. One more phosphorylation would take place. One more phosphate is attached. But right over here, this phosphate is inorganic phosphate. So right over here in this attachment, there is no utilization of ATP. Inorganic phosphate attached with glycerol 3 phosphate. So give us 1,3-bis phosphoglycerate. So as well as over here, we also give us one reduced NAD is produced as well. So NAD will convert into NADH2. So as a result of this, reduced NAD is produced, and one one inorganic phosphate is used up. So give us 1,3-bis phosphoglycerate. Now this 3 carbon compound would contain 2 phosphate molecules. So as a result of this, when 1,3-bis phosphoglycerate molecule would convert into 3 phosphoglycerates. So one phosphate is detached from the 1,3-bis phosphoglycerate. That phosphate is taken up by the ADP, adenosine diphosphate, to give us ATP. So right over here, one ATP is formed, right? So in the next molecule, so this reaction is, by say this reaction is cycled twice. So give us 2 ATP. Again over here now, this 3 phosphoglycerate would convert into 2 phosphoglycerate. This 2 phosphoglycerate would convert into pyruvate. So right over here again, one ADP is converted into ATP. So this, during the glycolysis of one glucose molecule, this process would be, revised twice. Bismillahirrahmanirrahim. Today we're going to discuss about the link reaction. आज हम link reaction को discuss करेंगे. So overall process during the link reaction would take place, that is pyruvate would convert into acetyl CoA. During glycolysis, each glucose molecule would give us 2 pyruvate molecules. So now these 2 pyruvate molecules would convert into the acetyl CoA, 2 molecules of acetyl CoA. So these 2 molecules of acetyl CoA later on used up in Krebs cycle. So there are 2 molecules of acetyl CoA, or we can say acetyl CoA is acting as a raw material for the Krebs cycle, so that it can be used up in the It can be used up in Krebs cycle or T.C.A cycle to give us further amount of energy. So to give us ATP, to give us FADH and NADH as well. So right over here, we say as a process of glycolysis, each molecule of glucose would convert into two pyruvate molecules to NADH molecule. We say to reduce NADH, to reduce NAD molecule and give us and also give us two ATP. Now we say as a result of so we say end product of glycolysis would be two pyruvate molecule, two reduced NAD molecule, and we say two ATP. Now these two pyruvate molecules they would move from cytoplasm to the matrix of mitochondria. So this movement from the cytoplasm to the matrix of mitochondria, it will take place in the process of active transport. Active, by the process of active transport, pyruvate molecule would move from the cytoplasm to the matrix of mitochondria. So inside the matrix of mitochondria, link reaction would take place. Sometimes you might have the question, what is the site of link reaction? The site of link reaction is matrix of mitochondria, which is inner core of the mitochondria. So in the mitochondria lecture, we would study about all the sections and all the regions of the mitochondria in detail. So right over here, so end product of glycolysis was pyruvate. So now this two molecule of pyruvate are produced. So these two pyruvate molecules, they would undergo decarboxylation, dehydrogenation, and also combine with coenzyme A. So right over here, in this process of link reaction, pyruvate decarboxylated. So during the decarboxylated from the pyruvate, carbon dioxide is removed. So during decarboxylation, this three carbon sugar would convert into the two carbon molecule. We say this three carbon molecule would convert into the two carbon molecule, which is acetyl CoA. So right over here, pyruvate is decarboxylated. As a result of decarboxylated, one carbon dioxide molecule is produced. So dehydrogenated as well. During dehydrogenation, reduced NAD is produced. Now this reduced NAD would be used up in oxidative phosphorylation to give us ATP because these reactions are the stepwise degrees of energy. Now we say this pyruvate also combined with coenzyme A. Coenzyme A is a very complex molecule. This coenzyme A is a complex molecule. It is made up of adenine and ribose as well. It is also made up as a mixture of adenine, ribose, and pentothenic acid. Pentothenic acid is vitamin B5 as well. So it's a pentothenic acid is one of the vitamin of B complex series, so which is vitamin B5. So this coenzyme A is acting as a carrier molecule for the transportation of pyruvate to this pyruvate is transport. Pyruvate is converted to acetyl CoA and is transported to the cycle which is known as Krebs cycle. So we say during the whole reaction, pyruvate would convert into the acetyl CoA. This acetyl CoA is transported to the T.CA cycle where it is further used up. So right over here, pyruvate combined with CoA plus NAD. So these are the reactant of this reaction. So as a result of this, acetyl CoA is produced. One carbon dioxide is removed. Carbon is removed in the form of carbon dioxide, which is acting as a waste product of this reaction as well. And reduced NAD is produced. So this reduced NAD would be later on used up in where it will use up in oxidative phosphorylation, which is also known as electron transport chain. So again over here during the fatty acid, during the fat metabolism, during the fatty acid oxidation, which is beta oxidation, also give us two carbon compound, which is acetyl CoA. Now this acetyl CoA also supposed to be entered into the T.CA cycle or Krebs cycle, where it is further processed as well, right? So our overall reaction of link reaction, I would say the overall process of link reaction is it would take place in the matrix of mitochondria. One thing. Other thing is so pyruvate is transported to the matrix through active transport where ATP is being used up. We say we use the word active transport. And the next thing is this pyruvate molecule is decarboxylated, dehydrogenated, and combined with coenzyme A to give us acetyl CoA plus carbon dioxide plus reduced NAD. So it's a reduced NAD would be used up later on in oxidative phosphorylation. We say over there it would give us energy in the form of ATP. We're going to discuss it later on as well. So this was the whole reaction of link reaction. So we say when one glucose molecule would undergo the link reaction, it would give us two acetyl CoA molecule, two reduced NAD, and plus like say also give us two molecule of carbon dioxide as well. When one molecule of glucose would accept, when one molecule of glucose would go under the link reaction. Thank you very much. Bismillahirrahmanirrahim. Our today's lecture is about the Krebs cycle or T.C.A cycle. This cycle was firstly described by the Krebs in 1937. So we use the word is a cycle because a closed pathway of enzyme controlled reaction is a closed pathway of enzyme controlled reaction. So we say during the aerobic respiration, glucose combined with oxygen to give us carbon dioxide, water plus ATP. So right over here, this one is known as cellular respiration. So this process is not this much of simple. It comprises of glycolysis. We have discussed already glycolysis. So as a result of glycolysis, pyruvate is produced. Now this pyruvate is used up in link reaction. We have already discussed it, about the link reaction. So link reaction would give us acetyl CoA. Acetyl CoA is a raw material for the Krebs cycle, right? So acetyl CoA is a raw material for the Krebs cycle so that the aerobic respiration should be completed. So we say as a result of link reaction, we will give us acetyl CoA. So this acetyl CoA would enter in the Krebs cycle to combine with oxaloacetate to form citrate, right? So we say acetyl CoA is combined with oxaloacetate to form citrate. Now if we see it over here, acetyl CoA is a 2-carbon compound and oxaloacetate is a 4-carbon compound. So this acetyl CoA, which is 2-carbon compound, would combine with oxaloacetate, which is 4-carbon compound, to form 6-carbon compound, which is known as citrate. Citrate is a 6-carbon compound which is formed with a combination of acetyl CoA, which is a 2-carbon compound, and oxaloacetate, which is a 4-carbon compound. Now this citrate would be decarboxylated and dehydrogenated to give us NADH and FADH, or we can use the word reduced NAD or reduced FAD. So right over here, citrate which is a 6-carbon compound, is decarboxylated. So during the reaction of decarboxylation of citrate, carbon dioxide is produced, and this during the process also citrate is dehydrogenated. So dehydrogenation of citrate would give us two molecules. One of them is known as reduced NAD, other one is known as reduced FAD. We can use the word reduced NAD, or we can use the word NADH2, or reduced FAD, we can use the word FADH2. So if we see it over here, now we have the 6-carbon compound, which citrate. The citrate was formed by the combination of 2-carbon and 4-carbon compound, which were oxaloacetate and acetyl CoA. So this citrate is decarboxylated. So decarboxylation process would give us FADH and NADH. Now we have oxaloacetate. So at the end of this reaction, at the end of this whole T.C.A cycle, oxaloacetate is regenerated, right? So we say at the end of Krebs cycle, oxaloacetate is regenerated. So this regeneration is mandatory for the continuation of Krebs cycle. So again, this in the next cycle, oxaloacetate combined with acetyl CoA to again give us citrate. Like this way, Krebs cycle will keep on continuing. We say as a result, when you say the end product of Krebs cycle will give us reduced NADH, reduced FADH, and it also gives us one ATP molecule. So this ATP is produced on substrate level phosphorylation. We already discussed it. 2 ATPs were also produced in, like say, during the glycolysis process. So total ATPs were produced. 4 ATP they were produced by the substrate level phosphorylation. So again over here in T.C.A cycle, 1 ATP is also produced by the substrate level phosphorylation. So we say one glucose molecule would give us 2 ATP molecule during the substrate level phosphorylation. Now we can say when we describe the overall reaction of Krebs cycle, so we say acetyl CoA, this acetyl CoA was produced during the link reaction. It was produced during the link reaction. Acetyl CoA would convert into acetate plus CoA. So acetate is taken up by the T.C.A cycle or Krebs cycle, and this CoA is removed from that T.C.A cycle, right? So we say acetate, which was known as acetyl CoA. Now acetyl CoA is removed, so left with acetate. Acetate combined with oxaloacetate. So acetate is a 2 carbon compound, oxaloacetate is a 4 carbon compound. So it would give us 6 carbon compound, which is known as citrate. We have already discussed this thing as well, right? Now, let's say this 6 carbon compound, which is citrate, is decarboxylated and dehydrogenated. In a first cycle, this 6 carbon compound, when it is decarboxylated and dehydrogenated, this citrate is a 6 carbon compound. Firstly, in the first reaction, it is decarboxylated and dehydrogenated, means during the decarboxylation, 1 carbon molecule is removed in the form of carbon dioxide. Citrate, 1 carbon molecule is removed from the citrate in the form of carbon dioxide. Now this 6 carbon molecule would left with, let's say it would remain with 5 carbon molecule, right? So this 5 carbon molecule, so first was 6 carbon is reduced with 1 carbon, and like say decarboxylated and dehydrogenated. So give us second molecule, which is 5 carbon compound. So now this 5 carbon compound is also decarboxylated. So it means 1 carbon is again removed in the form of carbon dioxide. So 5 carbon is decarboxylated and dehydrogenated as well. So we say decarboxylation and dehydrogenation, they would take place simultaneously. So in the beginning, we have studied that acetyl CoA is a, like say, raw material for T.C.A cycle. It combined with oxaloacetate, which was 4 carbon compound. So given a citrate, citrate is a 6 carbon compound. Now this 6 carbon compound citrate is decarboxylated and dehydrogenated. During the dehydrogenation and decarboxylation, 5 carbon compound is produced. Now this 5 carbon compound, from this 5 carbon compound, we would remove one more carbon in the form of carbon dioxide, and we also remove hydrogen in the form of NADH. So from carbon number, there is a 6 carbon compound and 5 carbon compound, one carbon dioxide is removed, and hydrogen, two atoms of hydrogen are removed to give us NADH in each reaction. We say, so it was in the beginning it was 6 carbon, it is converted to 5 carbon. Now 5 carbon is converted to the 4 carbon molecule. Now this 4 carbon molecule, we have first 4 carbon molecule. So this first 4 carbon molecule would convert into second 4 carbon molecule. First 4 carbon molecule would convert into second 4 carbon molecule. So it will so right over here for the during the conversion of first 4 carbon molecule to second 4 carbon molecule, a phosphate group is being added with the ADP to give us ATP. So right over here in the first reaction of 4 carbon molecule in T.C.A cycle, in the, like say, in the first reaction of 4 carbon molecule in T.C.A cycle, phosphorylation would take place to give us one ATP. So this ATP is produced over here again by the process of substrate level phosphorylation. Right? Now we say over here first 4 carbon compound convert into the second 4 carbon compound. Now this second 4 carbon compound would convert into third 4 carbon compound. Again, this second compound is 4 carbon, that is also 4 carbon, but right over here dehydrogenation will take place. So as a result of dehydrogenation of second 4 carbon compound to third 4 carbon compound, so dehydrogenation of second 4 carbon compound, it would convert the second to the third. So right over here hydrogen is released, so that hydrogen is taken up by FAD to give us reduced FAD or give us FADH2. Now we have, like say, this 4 carbon compound, firstly it would convert into again 4 carbon compound, give us one ATP. 4 carbon compound would give, like say, another, it would undergo another reaction to give us FADH. Now this 4 carbon compound would again convert into oxaloacetate. So over here, third 4 carbon compound, when it converted to oxaloacetate, so also hydrogen is removed, so that hydrogen is taken up to give us NADH, or we say give us reduced NAD or NADH2 molecule. We can use the word reduced NAD or NADH2 molecule. So this was the overall reaction of Krebs cycle. Now we can see over here. So during the Krebs cycle, 2 acetyl, let's say during the metabolism, one glucose molecule, 2 pyruvate molecule will produce. So these 2 pyruvate molecule will give us 2 acetyl CoA molecule, right? So each acetyl CoA molecule would give us how many? 2 ATPs, because this process will be repeated. So over here, it would also give us how many NADH molecules. So NADH was, firstly it was produced in the Like say in the first and second reaction and third reaction, three NADH molecule would also produce. Now we can say during the process of T.C.A cycle, it would give us three reduced NAD molecule, one reduced during each process, one reduced FAD molecule, and also give us one ATP. Now we say during the two when the two acetyl CoA molecule would use up to give us two ATP, two FADH, two, and like say we also give us during the two molecule will give us six NADH2 molecule. This was the end product of T.C.A cycle. So we can say during the end product T.C.A cycle, six NADH molecule are produced, two FADH2 molecule are produced, and two ATP molecule are produced, right? So over here, so whatever these NADH molecule and FADH molecule, they would be used up to form ATP, wherein the oxidative phosphorylation. So over there, the ATP produced from FADH and ADNDH in oxidative phosphorylation molecule, which is also known as oxidative phosphorylation reaction, which is also known as T.C.A cycle. Oxidative phosphorylation is also known as T.C.A cycle. So over there, FADH and NADH would use up, we can say reduce FAD and reduce NAD would use up to give us ATP, but that ATP is known as oxidative phosphorylation. But over here, so in the step, right over here, Bismillah ar-Rahman ar-Rahim. Today, we will discuss about the mitochondrial structure and function. So we say in eukaryotic cell, especially we are discussing mitochondria in eukaryotic cell because in prokaryotic cell there is no mitochondria. So in eukaryotic cell, mitochondria is a site for link reaction, T.C.A cycle, and oxidative phosphorylation. So as a result of glycolysis, pyruvate was produced, pyruvate was converted to acetyl CoA. So this acetyl CoA was formed in the link reaction, then this acetyl CoA which was formed in the link reaction sent to the T.C.A cycle. So right over here, this acetyl CoA again decarboxylated through the process of T.C.A cycles to give us NADH and FADH. So NADH and FADH were produced by the process of glycolysis, by T.C.A cycle and link reactions. So now they are used up in oxidative phosphorylation to give us ATP. So we say mitochondria is a site for link reaction, T.C.A cycle and oxidative phosphorylation. Now we say in each prokaryotic cell, they contain a mitochondria. So number of mitochondria depends upon the activity of the cell. So different cells of the tissues, so they have the different number of they have different number of mitochondria depending upon their activity. In case of hepatic cell, like liver cell, contain 1,000 to 2,000 mitochondria in each cell. So we say mitochondria occupy 25% of the volume of the cell. Now we say now we discuss the structure of the mitochondria. If you say function of mitochondria, including link reaction take place inside the matrix of the mitochondria, T.C.A cycle take place in the matrix of mitochondria, oxidative phosphorylation take place in the inner membrane of the mitochondria. Now we discuss the structure of the mitochondria. So mitochondria is a double membrane bound structure. So involved of the mitochondria is double membrane. So one of them is known as outer membrane and other one is known as inner membrane. So the composition and function of outer and inner membrane, they are absolutely different. Outer membrane is smooth and more permeable for the small molecule. Inner membrane folds to form cristae. So these cristae increase the surface area of the mitochondria. So the more multiple enzymatic reaction take place at the cristae, so increases the surface area of the cristae. We say the double membrane, inner membrane, and say outer membrane and inner membrane form cristae that increases the surface area of the surface area for multiple reactions. So at the inner membrane, electron transport chain or oxidative phosphorylation would take place as well. So synthesis of ATP also take place by the ATP synthesis enzyme which is present in the inner membrane. Now we say is the outer membrane of the mitochondria, then inner membrane. The central area of the mitochondria is known as matrix. So matrix is the region where link reaction, T.C.A cycle and oxidative phosphorylation would take place. And inner membrane, right over here, the inner membrane, proteins are embedded. They form the complexes of oxidative phosphorylation where electron transport chain is situated. So it would give us hydrogen ion gradients. So there is a hydrogen ion gradients between the like say between the matrix and intermembranous space. So intermembranous space is the area between the inner membrane and outer membrane. So because of this gradient energy is produced. So that energy is used up for the formation of ATP at the matrix by using ATP synthetase enzyme, by utilizing ATP synthetase enzyme. Now we say double membrane structure of mitochondria, outer and inner. So they have the different composition and function. So outer membrane is more smooth, inner membrane folds to form cristae. So this inner membrane folds to form cristae, and the number of the cristae depend upon the activity of the mitochondria. So these cristae are densely packed and they are large in number in more active mitochondria as compared to inactive mitochondria. We say active mitochondria would have more complexes, more cristae, which are densely packed. Densely packed, they are very close to each other. Now we say ATPs are formed in the matrix. So ATP synthetase are used for the formation of ATP. So hydrogen ion gradient between the inter and inner membranous outer and like say hydrogen gradient is formed between the matrix and intermembranous space. So hydrogen ions move inside through the ATP synthetase to give us ATP. So ATPs are formed at the matrix. Now we say we have the different multiple structure we can see inside the matrix. So one of the structure is known as ribosomes. So it also contains ribosomes. So these are 70s ribosomes which are present. These 70s ribosomes are present in the matrix of mitochondria as well. So mitochondria distinguish another structure as well. So it has its own DNA as well. It has its own circular DNA and its own mitochondria, like say its own ribosome. So we say mitochondria is known as semi-autonomous body. So we use the word semi-autonomous body because it can carry out certain of, like say it can fulfill partially its need as well. So we use the word semi-autonomous body for the mitochondria. Mitochondria has its own ribosome, which is 70th ribosome. So 70th ribosome makes it resemblance with prokaryotic cell. It has its own DNA. So this DNA is also similar to the DNA of prokaryotic cell. So there is an association between the mitochondria and prokaryotic cells. So that association was explained by endosymbiotic theory. Right? So endosymbiotic theory explains the origin of mitochondria, or we can say the evolution of mitochondria from the prokaryotic cell, right? So mitochondria has its own 70s ribosome in the matrix. It has its own DNA. So these two structures, mitochondrial outer membrane, also the porine. So these structures, like say, give us the clue that it was evolved from the prokaryotic cell. So in the beginning of the life, we say 3.8 million years ago. So there was only prokaryotic cell on the earth. So this prokaryotic cell developed to form a initial eukaryotic cell, which is known as primitive eukaryotic cell. So prokaryotic cell was evolved to form primitive eukaryotic cell. So when the size of this eukaryotic cell would, like say, keep on increasing, the size of the eukaryotic cell keep on increasing. So as a result of this, there the surface area to volume ratio of eukaryotic cell would like say word decrease. To overcome this issue, because by increasing the size of the cell, the surface area to volume ratio, which is concerned with the absorption and excretion of the cell. So surface area to volume ratio was reduced. To overcome this issue of surface area to volume ratio, because it was going to be reduced by increasing the size, this cell membrane folds to form small folds. So later on, these folds were pitched off from the cell membrane in containing the central DNA only. Containing the central DNA. So now this central DNA was surrounded by the folds, obviously surrounded by the small vesicles, which were pitched off from the cell membrane. So right over here, this primitive eukaryotic cell, it engulfed this primitive eukaryotic cell was a bit evolved or developed. This cell would engulf an aerobic bacteria. So as an engulfing of aerobic bacteria, so it, like say it start utilizing this aerobic bacteria for the formation of energy. Right? So when this energy was being formed with aerobic bacteria, So on the other end, this are also engulfed in cyanobacteria. So this cyanobacteria was providing the food for this bacteria, for this eukaryotic cell. So this eukaryotic cell, primitive eukaryotic cell, engulfed an aerobic bacteria. This aerobic bacteria modified to form is further, like say, modified to form mitochondria. So we say this mitochondria is evolved from the prokaryotic cell. So there are the certain points which, like say, which follow this theory, actually which support this theory. We can say this DNA of the mitochondria is similar to DNA of prokaryotic cell. It has a 70th ribosome, and this 70th ribosome is also present in the prokaryotic cell. And also, the very important thing, this, like say, mitochondria would multiply by the binary fission. So binary fission is also exhibited by prokaryotic cell. Another thing is that this membrane of the mitochondria, outer membrane, contain porins. So these porins are also present in the prokaryotic cell, in the membrane of prokaryotic cell.